Hello. Yes, my voice is audible. Please let me know. My voice is audible to all of you. Let me know. Yes, good afternoon to all. My voice is audible. Just waiting for students to join. Yes, how are you, Archana? Good afternoon to you. Yes, good afternoon to all of you. My voice is audible. Okay. So, what we have done yesterday? What is we just do yesterday? Please let me know. The topic which we covered yesterday. Any one of you, please let me know. The topic which you have covered yesterday. Please let me know. Protein. Okay, very good. So we have just covered protein. Have we completed the protein part or something is left? Have we completed it or something is left? Right. Please let me know. Have we completed it or something is there? तो मशीन का भी कुछ रहता है। तो आप घर वाले से बात कर लीजिए उनसे फ़ोन पे कि सर ऐसे बच्चे के थ्रू भी शुरू हो गया, प्रैक्टिकल नहीं शुरू होगा, घर वाले से पूछना पड़ेगा फिर। So have we completed the protein part? So yesterday what we have done, we have just started from here. So we will just start from there. Thank you. अरे वाह, पहली बार भी ले रहे हैं। हाँ 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 हाँ, थैंक यू सो मच, थैंक यू सो मच। कल मैं क्लास लूँगी, मैं टाइमिंग बताऊँगी, 11 से बजे लूँगी और थोड़ी लंबी क्लास लूँगी, ठीक है। बायो सिस्टमेटिक्स करेंगे ये वाली, और जो नया लड़का आया उसको भी बता देना, ठीक है। चलो, थैंक यू सो मच, टेक केयर। So yes, yesterday we have just completed with these things. We have covered enzymes. Let me show you. Yes. So we have done these things. We have done nucleic acid structure, structure of DNA, protein hormones, steroid hormones. Then we have covered enzymes. After it, lipid we have done. Fatty acids, phospholipid. This is your homework. We also done it. Then we are just there along with the protein part. So yesterday we have done protein, the structure of a proteins basically. So these all the 3 days we have to do proteins only. So let me share you the PPT. So because protein part is very important, let me show you the presentation. I think we have not completed yesterday. Some of the part is left. Let me check it. So we have done, yeah. वो बच्चे को कहो कि सीओई को एप्लीकेशन लिखे और एचओडी को, हाँ मुझे सीसी कर दो। हाँ उसको कहो कि सारी जो भी उसकी प्रॉब्लम है एवरीथिंग, पहले उसकी एप्लीकेशन चेक कर लेना, क्या आपको पहले व्हाट्सएप पे भेजेगा। आप तो एक बार गो थ्रू करके ना सैमसंग सही डाला, फिर हम उसको सीओई को कर दे, मुझे सीसी कर दे साथ में। So we have just done this part before also. This is the protein hormone basically. After it we also cover the protein, the structure of a protein, the functions of that particular protein. Let me open it. So we have already discussed all these things, but we have not completed it. So what is the homework which I told you yesterday? What is the homework which I told to all of you to complete? Anyone, please let me know. Yeah, like this GABA is there, we have done this GABA. Okay. So what is the homework which we have done yesterday? Any one of you, please let me know the homework. Have you completed your homework? Okay. So we have to complete amino acids based on their structure. Okay, okay. So we have covered amino acids. Properties of amino acids. Okay. I think these are the amino acids which I told you yesterday to complete, right? You have to do all these amino acids according to the on the basis of their structure. So this is the classification of the amino acids. Yes. So I told you to do the classification of amino acids along with the classification of the amino acid according to their structure. Now I will continue from here only, okay? So according to the structure, the amino acids are basically aliphatic amino acids. So now I will continue from here. So are you getting me? My voice is audible to all of you? Okay, so shall we start now? Shall we start now? Okay. So now I am just going to start it. Okay, Suradeepa, I will definitely check my mail, but I will reply to you only by this weekend. That is by Saturday or Sunday, I will able to reply to you because I am quite busy right now. So I will try to complete it after the. I will try to reply you during the weekends. Okay. So no worries, I will check it. So now I am just going to start. So this is the classification of amino acids which we have already done yesterday also. This is the classification on the base of their structure. So on the base of the structure we have 7 groups of amino acids. I hope you all have done the homework also. So we have 7 distinct groups. We have aliphatic amino acids, hydroxyl group containing amino acids, sulfur containing amino acids, acidic amino acids, basic amino acids, aromatic and amino acids. So these are the amino acids on the basis of their structure. So this is also your homework. I hope all of you have done it. So this is the aliphatic amino acids. So these are the monoamino monocarboxylic acids. This group consists of simple amino acids. So in this group we have glycine, alanine, valine, leucine and isoleucine. So the glycine is a simple small amino acid group. It is a non-essential amino acid. Glycine is allosteric inhibitor of glutathione synthase. So alanine is again a non-essential amino acid. Alanine is an allosteric inhibitor of glutathione synthase. So we have two types of alanine. We have D-alanine which is found in bacterial cell wall and we have B-alanine which is found in pantothenic acid. Again we have valine, leucine, isoleucine, hydroxyl group containing amino acids, serine, threonine, sulfur containing amino acid which is cysteine and methionine, cysteine which is a non-essential amino acid. Cysteine, another important sulfur containing amino acid, is formed by the condensation of two molecules of cysteine. Then we have methionine, then we have some amino acids with acidic charge. We have aspartic acid, glutamic acid. We already discussed it yesterday also. So I am just going to repeat it here. So then we have basic amino acid which is basically lysine, arginine and histidine. After it we have aromatic amino acids, in which we have a ring. So we have phenylalanine, tyrosine and tryptophan. So this is the phenylalanine, tyrosine, tryptophan. After it we have amino acids. So this is the classification of amino acids based on the polarity. So first we have the classification on the basis of structure. Now we have the classification on the basis of polarity. So according to the polarity, the amino acids can be classified into 4 groups. So we have 4 groups according to the polarity. So we have non-polar amino acids. These amino acids are also referred to as hydrophobic. Then we have polar amino acids with no charge. After it we have non-polar amino acids with positive R group. So here we have polar amino acids with no charge. So these amino acids are as such carry no charge on the R group. They however possesses groups such as hydroxyl, sulfhydryl and amide and participate in the hydrogen bonding of protein structure. The example is glycine, serine, threonine, cysteine, glutamine, asparagine and tyrosine. After it we have polar amino acids with positive R group. Now we have the polar amino acids with positive R group. We have lysine, arginine and histidine. So polar amino acids with negative R group. So the carboxyl monoamino acid, aspartic acid and glutamic acid are considered in this group. After it we have classification on the basis of nutritional value. So according to it we have essential or indispensable amino acids. So the amino acids which cannot be synthesized by the body and need to be supplied through the diet are called essential amino acids. They are required for proper growth and maintenance of the individual. So we have essential amino acids. So we have arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine and tryptophan, which is the essential amino acids. Similarly, we have some non-essential amino acids also. So the bodies that cannot synthesize these 10 amino acids to meet the biological need, hence they need to be consumed in the diet. So these are glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, glutamine, tyrosine and proline. So these are the non-essential amino acids. Then we have based on their metabolic fate also. So the carbon skeleton of amino acid can serve as a precursor for the synthesis of glucose or fat or both. From metabolic viewpoint, amino acids are divided into 3 groups. Now the very first group is glycogenic amino acids. So these amino acids can serve as a precursor for the formation of glucose or glycogen. The very common example is alanine, aspartate, glycine and methionine. So these amino acids, these alanine, aspartate, glycine and methionine, these amino acids can serve as precursor for the formation of glucose or glycogen. Similarly, we have ketogenic amino acids. Fat can be synthesized from these amino acids. So leucine and lysine are basically ketogenic in nature, from which we will get the fats. So these are both glycogenic as well as ketogenic. So this is a thing we have already done yesterday also. Now the main thing which we have to do today is properties of amino acids. Are you getting me? Yeah. So the main thing which we have to do right now is the properties of the amino acids. Because we have done the structure of amino acid, the classification of amino acid according to their polarity and everything. Now we have the properties of amino acids. So solubility. So most of the amino acids are soluble in water and insoluble in organic solvents. So the amino acids are basically water soluble. Now the melting point. So amino acids generally melt at higher temperature, often above 200 degrees Celsius. So amino acids melt at higher temperatures. So it needs up to 200 degrees Celsius for proper melting. Then taste. So amino acids may be sweet, tasteless or bitter. Monosodium glutamate is a salt of glutamic acid. It is employed as a flavoring agent in food industry to increase the taste and flavor. So optical properties now. So this is the solubility, melting point, taste and the flavoring agent. It is used also in the flavoring agent in the industries to add the taste and flavor. After it we have optical properties. Now what are basically the optical properties? So all amino acids except glycine possesses optical isomers due to the presence of asymmetric carbon atoms. So we have the optical isomerism because of the presence of asymmetric carbon atoms. So if you will see here, amino group is attached here in case of L amino acids. In D amino acid, amino is on the another side. So amino acids as ampholites. So amino acids contain both acidic and basic groups. They can donate a protein or accept a proton. Hence, they are also known as ampholites. So they can donate the protein as well as they can accept a protein. So they are also known as ampholites. Now the very another common term which is Zwitter ion or dipolar ion. Now what is basically Zwitter ion? So Zwitter ion is a hybrid molecule containing positive and negative ionic groups. So in the Zwitter ion we have the positive as well as negative ionic groups. The amino acids rarely exist in the neutral form with free carboxylic or free amino groups. In strongly acidic pH, the amino acid is positively charged. In strongly alkaline pH, the amino acid is negatively charged. So, Zwitter ion is a hybrid molecule containing positive and negative ionic groups. Okay. So, Zwitter ion. So, each amino acid has a characteristic pH at which it carries both positive and negative charge and it exists as Zwitter ion. So, when the amino acid at a particular pH, any amino acid at a particular pH, when it carries both positive as well as negative charge, it is known as the Zwitter ion stage. So, amino acid has a characteristic pH at which it carries both positive and the negative charge. That state is known as Zwitter ion. Then after it we have isoelectric pH. Now what is isoelectric pH? It is defined as the pH at which a molecule exists as a Zwitter ion or dipolar ion and carries no net charge. So isoelectric pH is defined as the pH. So you have to keep in mind these terms. We have Zwitter ion, we have a isoelectric pH. So isoelectric pH is the pH at which a molecule exists as Zwitter ion or dipolar ion and carries no net charge. So there is no positive or negative charge. Now these are some physical properties of amino acids. Now after it we have some chemical properties of amino acids. So the general reaction of amino acid are mostly due to the presence of two functional groups, namely carboxylic and amino group. So we have two types of reactions which will occur. So reactions due to COOH group. So amino acids form salt COONA with bases and esters with alcohols. So we have a few reactions which is because of this COOH group. So decarboxylation. So amino acids undergo decarboxylation to produce amines. So histamine, tyramine, GABA. From the amino acid histidine, tyrosine, glutamate. So from the amino acid, what we will get? We will get the amine. We will get the amine. So from histidine, we will get histamine. So this is because of the COOH group. What we will get? We will get the amine. So this is known as decarboxylation. Now what is basically decarboxylation? When from the amino acid we will get the amine. Then we have reaction with ammonia. Now when these acids, which is COH, it reacts with ammonia, it will lead to the formation of abide. First we have the formation of amine, now we have the formation of abide, and from aspartic acid what we will get? We will get the asparagine. For glutamic acid what we will get? We will get the glutamine. So then we have the reactions due to the NH2 group. So the amino group behave as bases and combine with acids to form the salt. So this is the reaction which is due to the NH2 group. So now the another is reaction with ninhydrin. Now what is basically ninhydrin? The amino acids react with ninhydrin. This is again the chemical reaction or chemical property. So the amino acids react with ninhydrin to form purple, blue or pink color complex. So what we will get? We will get purple color, blue color or pink color complex, which is known as Ruhemann's purple. So this is the reaction of the amino acid with the ninhydrin. What we will get? We will get the colored complex. So what we will get? We will get the colored complex with the reaction with the ninhydrin. So these are the some reactions of these amino acids, and when there is a reaction we will get the specified color. We will get the specified color. So we have biurate reaction. This is also the reaction of amino acids. So from biurate we will get two peptide linkages. Ninhydrin reaction we will get alpha amino acids. So all these are the tests which are specifically for specific amino acids. If you are able to see, we have millions reaction which is basically for the phenolic group, which is tryptophan. Similarly, we have sulfur test for cysteine. We have Pauline's test for histidine. We have Pauline's test for tyrosine. So we have different reactions for different amino acids. Then we have another term which is transamination. Now what is basically transamination? The transfer of an amino group from an amino acid to a keto acid is to form a new amino acid is known as transamination. Now what is basically transamination? It is the transfer of the amino group from amino acid to the keto acid. So from amino we will get the keto. So this is transamination. Then we have another term which is oxidative deamination. Now what will happen in this? The amino acids undergo oxidative deamination to form free ammonia. So what we will get basically from oxidative deamination we will get the free ammonia. You have to keep these points in mind: transamination, oxidative deamination, but because we have to discuss it in detail also. Okay, so decarboxylation, transamination and oxidative deamination, we have to discuss their metabolic paths also. Now we have amino acids drive in proteins. So the 20 standard amino acids can be incorporated into the proteins due to the presence of universal genetic code. Some of these amino acids undergo a specific modification after the protein synthesis occurs. These derivatives of amino acids are very important for protein structure and function. So we have the very first one is collagen. Now what is collagen? The most abundant protein in the mammal contains 4-hydroxyproline and 5-hydroxylysine. The most abundant protein in mammals contain 4-hydroxyproline and 5-hydroxylysine. Then we have histones. So these are also the proteins which are associated with the DNA. Getting my point? So these proteins are associated with the DNA, contain many methylated, phosphorylated and acetylated amino acids. So we have alpha carboxyglutamic acid is found in certain plasma proteins involved in blood clotting. So cysteine is formed by the combination of two cysteines. It is also a derived amino acid. So these are some non-protein amino acids which we have. These are ornithine, citrulline, arginosuccinic acid. These proteins are non-protein in nature. These amino acids are non-protein, but yes, they are very important in the some biochemical reactions. So we have thyroid, which is thyroxine, T3, T4, homocysteine, keratin. So arginine is very much important in the synthesis of urea. Citrulline is important in the synthesis of urea. Arginosuccinic acid is intermediate in the synthesis of urea. Similarly, keratin is there, which is for the excretion in the urine. So these are some amino acids which are not proteinaceous in nature, but they have very important role to play. So these are non-alpha amino acids. Now we have another term which is non-alpha amino acid. Now what is basically this non-alpha? So we have B-alanine, B-amino isobutyric acid, alpha aminobutyric acid, which is GABA, then delta aminolevulinic acid, taurine. So they all have some functions. So these are non-alpha amino acids. That means alpha is absent. Rather we have beta or delta. So the component of pantothenic acid and coenzyme A, so they have their own functional activity. So you have to keep in mind all these amino acids which may be proteinaceous in nature, which may be non-proteinaceous or without the protein. So you have to keep all these in mind. After it we have D amino acids. Now what are the D amino acids? Certain D amino acids are also found in the antibiotics. D-glutamic acid and D-alanine, they are present in the bacterial cell wall. D-serine and D-aspartate are found in the brain tissue. So we have D-alpha amino acids. So up to it we have amino acids. After it we will have to move forward to the peptides and proteins. So up to now, is it clear to all of you? Up to now, is it clear to all of you? Please let me know. Because we have to discuss protein part also. So we have done amino acids and proteins, essential and non-essential amino acids we have done. Now we have another term which is the proteins. What are the structure of the protein, their functions and their properties also. So now we will move forward to the proteins because we have done amino acids right now. So shall we continue? Shall we continue? Please reply. Please reply, any one of you. Hello, my voice is audible. Okay, so now after the amino acids we have to start the protein part. Okay. So now we have to start proteins and peptides. So what are basically proteins? So 20 amino acids which we have. So 20 amino acids, they are commonly found in the proteins. The 20 amino acids basically are linked together through the peptide bond forming peptides and proteins. So these amino acids which we have, they will link together and they will form the peptides and proteins. So the chains containing less than 50 amino acids, they are called peptides. When we have more than, or when the chain is greater than 50 amino acids, then it is known as protein. So we have peptides as well as we have proteins. Now peptide bond formation. The alpha carboxyl group of one amino acid forms a covalent peptide bond with alpha amino group of another amino acid by removal of a molecule of a water. The result is dipeptide. So what we will get basically? Dipeptide. Alpha carboxyl group of one amino acid forms a covalent peptide bond with the alpha amino group of the another amino acid. So by which we will get the dipeptide. So the two amino acids, they are linked by peptide bond only. So the dipeptide can then form the second peptide bond with the third amino acid to give the tripeptide, and so on we will get the polypeptide, or we will get the protein of specific amino acid sequence. So we will get the protein of specific amino acid sequence. So each polypeptide chain starts on the left side by free amino group of the first amino acid and then in a chain formation. After it is termed as N-terminus. Each polypeptide chain ends on the right side by free COH group of the last amino acid, and it is termed as C-terminus. So we have N-terminus, we have C-terminus. So each polypeptide chain starts on the left side by free amino group of the first amino acid entered in a chain formation. It is termed as N-terminus. So we have N-terminus as well as C-terminus. Now we have peptide bond. The amino acids are held together in a protein by covalent peptide bonds or linkage cementing material between the amino acids. So when amino group of an amino acid combines with the carboxyl group of another amino acid, a peptide is formed. So we have one amino group and on the other side we have the carboxyl group. So they will join together by the peptide bond. So peptides containing more than 10 amino acids, it is known as polypeptide. So this is a thing. One side we have the carboxyl group, on the other side we have the amino group, and they will join together by the peptide bond. So they will join. So this is a function of polypeptides. It is usually found in trans configurations. The peptide bond is a partial double bond, N partially positive, O partially negative. Shorthands to read peptides. The amino acid in a peptide or the proteins are represented by the 3-letter or 1-letter abbreviation. This is the chemical shorthand to write the peptide. So this is basically one side it is partially positive and one side it is partially negative. Now how after amino acids, now we have to work on peptides. How we have to add the suffix in peptides. So we have glycine, tryptophan, glutamate, they are changed into "-ile". When we are working in the peptides, then they are changed into "-ile". They are changed into "-ile" with an exception of C-terminal amino acid. So what will happen here? Glutamate will change into glutamyl. Similarly, cysteine will change into cysteanyl. Then glycine will remain glycine, but otherwise all will change into glutamate. Glutamate will change into myl. Cysteine will change into cysteanyl. So this is the how we are able to name the peptides. Now the dipeptide, two amino acids are joined together by one bond. The example is aspartame, which acts as a sweetening agent being used in the replacement of cane sugar. So here what happens, it is composed of aspartic acid and phenylalanine. Tripeptides, we have 3 amino acids linked by 2 peptide bonds. So this is how we have to name the peptide. So instead of ine or an, it will changes into YL. So it is only here glutamyl. Then again we have another example which is formed from 3 amino acids, which is GSH. It is glutamic acid, cysteine and glycine. It helps in the absorption of amino acids, protects against hemolysis of RBCs by breaking H2O2, which causes the cell damage. So similarly we have insulin, which is also a polypeptide hormone. So it is also made up of 10 to 50 amino acids. So 10 to 50 amino acids, they will join together and they will form the polypeptide insulin. After it we have structure of proteins. So up to now, is it clear to all of you? Then we will move further to proteins. Up to now, is it clear? मैं तो ले रही हूँ क्लास. आप लोग बात करनी है या फिर 4 बजे तक रुक जाओ. 4 बजे मेरी क्लास खत्म होएगी. ओके. मैंने बस अभी हेडफोन ही निकाला. हमारे तो नहीं लेके जा रहे कि देखो कैसे बच्चे हैं, ऐसे बच्चे हैं ये. जा सकते हैं, देखते तो है ही नहीं. देखते हैं कहाँ पे लेके करना है ये तो बताओ. फिर देखते हैं. So up to now, is it clear? Shall we move further to proteins? Please let me know. अच्छा जसमीन, मुझे पानी भर के ला के देना नीचे से. एक बच्चा कोई भी लिया. एक मिनट. So this is basically the proteins which we have to study right now. Yeah. So this is the structure of protein. We have 3 different levels of organization of the proteins. We have primary structure, secondary structure and tertiary structure. So primary structure is a linear sequence of amino acids forming the backbone of the protein. The secondary structure is the spatial arrangement of protein by twisting of the polypeptide chain. Then we have the tertiary structure which consists of functional protein. And the last is the quaternary structure which will be definitely little bit more complex than the previous ones. So in the quaternary structures we have 2 or more polypeptide chains which will join together. So if you are able to see here, the primary structure is a very simple one. After it we have the secondary structure which may be alpha helical or beta plated. Then we have the tertiary structure. In the quaternary the 4 subunits are joined together. So in case of quaternary the different subunits are joined together and it is known as quaternary structure. So up to now, is it clear to all of you? Please let me know. बैठ जाओ 2 मिनट, बस खत्म होने वाली है. So if you have any query, please let me know. Any query regarding the proteins. तो इसमें क्लिनिकल में ले लो. हाँ सर. नहीं ना, अगर यहाँ आना तो मैं तो क्या रहती है तो कब तक जाएंगे. तो हाँ बिल्कुल बिल्कुल. थैंक यू. अभी हमें जाना है, फीस का हुआ नहीं ना. कब आओगी फिर? मंडे आओगे? कल मंडे में आना. कल ऑनलाइन है सारी कुछ. कल मतलब मंडे आना. मंडे आना क्योंकि कल तो फिर बाद में किसी ने भी नहीं आना, बच्चों ने भी नहीं आना. नहीं वो ऑफलाइन है ना, ऑनलाइन नहीं भाई. ये तो बेचारी बहुत काम करती है. वो ऐसे ही आपको रिसीव कर लेगी पर ऐसे काम है भी यार का फोन करके सुबह 6 बजे बताना कि आज ऑफलाइन में है ऑनलाइन. अच्छा चलो फिर आप सोमवार आना. ठीक है. ठीक है बच्चे. मंडे मिलते हैं. मेरी क्लास करिए. मंडे मिलते हैं या फिर 4 बजे तक रुक जाओ. 4 बजे बात करूँगी. हाँ फिर आ जाना. फिर 4 बजे आ जाना. Okay. So it is clear to all of you. Please reply. Why you are not replying to me? You have to let me know whether you are getting it or not. The protein structure is clear to all of you. Shall we continue now? Archana, Mudita, shall we continue now? So yes, so we have this quaternary structure. So we have primary structure, secondary structure, tertiary structure and the quaternary structure. So primary and the secondary are the simple ones and tertiary and quaternary structures, they are the complex one. Okay, then we have some structure of protein which we have to discuss again. We will start with the primary structure. The linear structure of amino acid held together by the peptide bonds in its peptide chain. So the peptide bond forms the backbone. The free NH2 group of the terminal amino acid is known as N-terminal and the free COH is known as the C-terminal. So where we have the NH2 group attached, it is known as the N-terminal, and where we have the COH group attached, it is known as the C-terminal. So you have to keep this thing in mind. So it is a tradition to the number the amino acids from N-terminal end as number 1 towards the C-terminal end. The presence of specific amino acid as a specific number is very significant for a particular function of a protein. So this is a very simple structure of primary proteins or the primary structure. We have only amino acids which are joined together by the peptide bond. So this is alanine, methionine, lysine, arginine, histine, leucine, tryptophan. So they all are joined together with the help of the peptide bonds. And at the end we have the C terminal where we have the carboxyl group attached, and we have the N terminal where we have the amino group attached. So this is determination of the primary structure. So the primary structure comprises identification of amino acid with regard to their quality, quantity and sequence in a protein structure. So determination of primary structure involves 3 stages. We have amino acid composition, which is we have to do the determination, then we have degradation of proteins or polypeptides, and at the end we have the determination of the amino acid sequences. So we will discuss this part. I think that's all for today. We will discuss it later. So for tomorrow we will have to do the determination of the primary structure, which involves 3 stages. So we have to discuss these 3 stages in detail. Determination of amino acid composition, degradation of protein and determination of the amino acid sequence. So I think that's all for today. The remaining part we will discuss later on. So yes, yeah. So I think that's all for today. Yes, now the homework which you all have to do, you have to let me know about the protein structure, the primary structure, secondary structure, tertiary structure and the quaternary structure. So you have to tell me about these structures along with the diagram. So is it clear to all of you? Please let me know. Please let me know if the homework is clear to all of you. Okay, good, Pooja. So any one of you have to type the question also. So is the question is also clear to all of you? Please let me know. Please let me know. Yeah, Mudita, please let me know. So I just keep it very simple today. We will discuss it later on because the protein part is very important. So I just focus today on the amino acids, peptides as well as the structure of proteins. We will discuss it later. Please any one of you just let me know whether the question is, whether the homework is clear to all of you. Please let me know. Okay. मैंने करवाना मैंने तो करने में कर दिया इसको जाओ वो तो सब्जी वो हो गया सर सर्दी में कर दे वो ठीक है वो तो मुझे कोई बात नहीं वो तो कोई बात नहीं दिमाग पता नहीं मैं सर के लिए दवा लेती हूँ फिर अपना दिमाग Yes, yes. So you have to tell me about the protein structure. You have to draw as well as you have to write it also. So this is your assignment. So I hope any one of you please just pin this also so that all will get the notice also. So that is all for today from my side. We will meet tomorrow. Till then, take care of yourself. ठीक है।
टोटल 24 तो बच्चे बन रहे हैं। बीएससी की एक ही लड़की शेष है, वो भी पता नहीं चाहिए ये नहीं जाएगी।
एक तो अफाक उमाइस का आखिरी हो गया
वो हो गया
ना बीएससी के वो हाँ है ना तीनों अफाक आकिब और उमाइस का तीनों यार उन्हीं को तो उनको भी मैंने प्रोजेक्ट दिया था, वो भी कश्मीर से आए हुए हैं, तीन लड़के 5th सेम किया, फिर 10 नहीं वो थर्ड सेम किया, सॉरी थर्ड सेम किया।
मेरे को तो मतलब सिमरन है, अभिषेक है,
मेरे पास तो 3 बच्चे हैं थर्ड सेम के, जिसका मैं एग्रीकल्चर का देखती हूँ ना क्लास, इसलिए उनको भी लेके जाना है। और फिर टेंजिला थर्ड वो बच्चा है उसका कोई उनके साथ कोई बच्चा हो। फिर आखिरी वो 3 का उनका लिख लो, वो भी बीएससी के ये तो है ही नहीं।
ये कह रहे हैं ये तो भी मेरे को मिली ही लड़की, अभी इनकी प्रैक्टिकल की कोई रुचि तो मैं बोल के नहीं कर रहा था, बोर्ड मैंने पूछा बोलते हमारे बॉटनी में थोड़ी भी है
जब बीएड वाले होंगे भी पता नहीं, मैंने नामी पहली बार सुना है। और एक बच्चा बीएससी फर्स्ट में आया, उसका नाम नासिर है, वो भी आया हुआ है बच्चा, देखो मैं आपको बताती हूँ। सो दैट्स ऑल फॉर टुडे, प्लीज टाइप द questions, okay? So take care of yourself. If you have any query you can ask me, any one of you, if you have any query please let me know, all of you please let me know if you have any query. Please ask me if you have any query, otherwise we will continue it tomorrow also. And try to submit your assignments on time, okay? So please keep it on time. ठीक है, जो थोड़े से बच्चे हैं 30 में लगा के कर लेते हैं, तभी फिर मैंने कहा रजिस्ट्रार के पास ले जाऊँगी कि मैं तो बच्ची बहुत छोड़ हूँ। और एमएससी में है 4 बच्चे, हाँ 4 वो
अब बीएससी बताओ, बीएससी किस सन में कौन
3 है, नहीं थर्ड में
बताओ
आकिब, अफाक, उमाइका।
और किसमें से अब नहीं था, तुम ये कह रहे हैं हमारा भी लगा वाला
ये तो मैंने
अभिषेक को
मैं दिल्ली क्लास में ले गई। अब हम देखेंगे कभी बच्चे, देखेंगे कभी हमें नहीं ना वैसे के बाद हम पहले भी देखेंगे।
गवर्नमेंट आपने देखा हमने उनको बताया कर दूसरा हमने कर लिया तो कैसे लगेगा
तो रख लो लिख लो, कोई बात नहीं, पर मुझे पता नहीं है।
मैंने पूछा, मैंने प्रश्न पूछा कहते हाँ हमारा जूलॉजी भी है, हमारा बॉटनी भी है,
तो फिर रख लो कोई नहीं।
इसका नाम कुछ बीमार थी, एक लड़की बीमार थी, लगाने की पुराने ही देते हैं तो मैं अभिषेक दोगे, अब शेष तक 6th टाइम की मुझे लगता है वो भी जाने वाली, मुझे उसने जाना ही नहीं। हाँ, ध्यान दो। ये हो गए 8